George, for that really kind invitation and introduction. So, as I said, Amy and I are here from Liverpool, and we're going to be giving a bit of background on the in vivo mouse work that we do as part of the Centre for Snakebite Research and Interventions, and also on a wider background of other neglected tropical diseases. There we go. So today's seminar it's a long seminar. We've got 2 hours. We're going to fill it out. So we're going to do an introduction to the 3 Rs. We're going to give you a bit of background onto the UK animal research regulation, and also give you a bit of background on how we go about mouse welfare in the UK. We're going to give you, Amy's then going to give you an example of the 3 Rs in NTD research. And she's also going to give you an example of how we are refining the current gold standard envenoming models for preclinical snakebite research. And then we're going to finish up with me just giving you a bit of a talk on the current state of preclinical testing of antivenoms for sub-Saharan Africa. Okay. If you have any questions at any point, please feel free to put your hand up. If you need anything clarifying, again, feel free to put your hand up, and I'm happy to do so. So, on top of the introduction that George just gave you, I want to give you a bit of background to demonstrate, to prove that we do have a bit of experience in this field. So myself, I'm a research fellow and a lecturer at CSRI. I've had a personal license. Now, personal license we'll go into a bit more detail later, but personal license is a document that allows you to carry out animal research in the UK. So I've had a personal license for 7 years. I've had a project license, which is another license we'll go into. It allows you to oversee a body of animal research. I've been a project license holder for 5 years, and I'm also the deputy chair of the Liverpool School of Tropical Medicine's Animal Welfare and Ethical Review Board. Amy is a postdoctoral research associate. Very sorry for the spelling mistake. Amy has a wealth of experience in animal research. She's had a personal license for 8 years. She's been a member of the LCMA work for a very long time. She's got a lot of experience with mice and gerbils. She's worked on over 4 disease models, mostly tropical diseases. She's proficient in greater than 10 regulated procedures, and we'll talk about regulated procedures in a minute. And she also is one of the rare few people in the UK that has animal experience in CAP3 laboratories as well, working on COVID during the pandemic. So we've got a lot of background. So, mouse welfare. First question, why should we improve mouse welfare in research? Well, it's quite simple, really. Mice do feel pain and distress. Mice and humans, we share lots of the same anatomical and pathological, physiological pathways. We show similar behaviors, physiological behaviors in responses to painful stimuli. And we also use mouse models, as pain models for research into human pain. So they do feel pain and distress, and we should recognize that and try and reduce it as much as possible. And there's 2 types of pain that the mice feel, really. So there's the acute pain, which is short-lived and not necessarily severe in nature. You can see this apparently quickly when you give a noxious stimuli. So if you give them some venom, they show signs of acute pain very, very quickly. And you can see this by changes in behavior, sort of vocalizing, they're reluctant to move, they can be irritable, abnormal posture, but they can respond quite well to analgesics or removing of that stimuli. The other pain is chronic, so this is more long-term pain. It can be difficult to detect, and it requires careful observation. It can show through decreased appetite, weight loss, reduced activity, sleep loss, and irritability, and it can be difficult to treat and difficult to detect. So, knowing that mice do feel pain in a similar way that we do, why? On top of that, another reason that we should improve animal welfare is that there is a lot of evidence, there's a wealth of evidence now that suggests that pain and suffering can alter an animal's behavior, physiology, and its immunology. And these are all aspects that are key, the variables that are key in a lot of animal research. You do not want this pain and distress actually affecting the experiments that you're doing and the research you're doing, because these can lead to variation in experimental results from experiment to experiment, and it does impair the reliability and the reproducibility of studies. So in short, you should improve mouse animal welfare, because poor welfare leads to poor science and wasted resources, okay? So at the very end of the day, you don't want to do poor science, you don't want to waste money. So you're going to hear us talk a lot about the 3 Rs, and I hope a lot of you have heard of the 3 Rs before. So the 3 Rs are the replacement, reduction, and refinement of animals in experimental research. And this was developed in the 1950s. And since then, it's been embedded in national and international regulations and in many countries into legislation for the use of animals in scientific research. And not only that, it's also been embedded in sort of the procedures and policies of funding bodies that fund such research as well. So the 3 Rs are of immense importance. I'll just go through them quickly. So replacement is the first one. So replacement refers to technologies or approaches, methods which directly replace or avoid the use of animals in experiments where they would have otherwise been used. So we have two types of replacement. There's full replacement, where we can have a complete avoidance of animals in research. So this is frequently these days, we're moving towards human challenge models. We can use tissues, we can use cells. Here we've got organs on a chip, which are starting to generate a lot of excitement for replacing animals in early preclinical studies. Or we could use mathematical models. If you can get very good variability data and do some good mathematical modeling on your pharmacological properties, you can replace the animal altogether. We then have partial replacement, where we're using animals that are not considered capable of experiencing suffering. So this is something that is clearly defined in the UK legislation. So it could be invertebrates, so Drosophila, nematodes, amoebae, or immature forms of vertebrates. So in the UK, nothing's considered an animal until 2/3 gestation, basically. So we've frequently started using ovo models. So if you've got a fertilized egg, that fertilized egg will develop a lot of physiological traits. It will have a heartbeat, it will have a circulation. You can do experiments on that up until 2/3 gestation. Reduction. So reduction is what it says on the tin, methods that minimize the number of animals used per experiment. And this isn't just reducing the number of animals overall, it can be reducing the number of animals throughout the course of a study. So these are methods which allow the information to be gathered per animal in an experiment to be maximized. So this is a bit more complicated. So you can use, for example, we've got two examples here. We've got imaging modalities. So this allows basically repeat observations on longitudinal studies in the same animal to be taken. So you don't have to cull a mouse, essentially, lots of mice at lots of different time points to get the amount of material you need to process for your experiments. Another thing is microsampling of blood, where you can take repeat very, very small amounts of blood temporally over a time course from an animal. So you can monitor responses to drugs or pharmacokinetic profiling. And this is something that we're looking at quite a lot at the moment in our envenoming models for coagulopathic pathologies. And another thing reduction could be is simply sharing data and resources. If you have animal tissues, animal data that you are not requiring in your collection, but another laboratory needs it for their research, instead of that laboratory consuming animals to get that data, you can simply provide that data to them, thus using smaller numbers of animals. And then finally, refinement. Refinement, in my opinion, is the most important. So this is methods that minimize the pain, suffering, and distress, or lasting harm experienced by research animals, and improve their welfare. And it applies to all aspects of a mouse's life, from their housing and their husbandry, all the way through the scientific procedures that produced on them. And it could be as simple as an improvement in a refinement to an animal model, or just animal husbandry, is allowing mice to have nesting material, so they can demonstrate normal behavioral species-specific behaviors. Or it could be something on the other end of the spectrum, making sure you're using appropriate anesthesia, so there are appropriate planes of anesthesia before you start surgery, or you're using appropriate analgesia. Your analgesia is appropriate for the interventions that you're doing. So you're not giving paracetamol, but you're injecting them with venom. You're going to need something stronger like morphine or tramadol. So I'm going to give you a bit of an overview how in vivo experiments are regulated in the UK, because it's very, very, very strict. So in the UK, all animal scientific procedures are regulated under the Animal Scientific Procedures Act 1986, which has been updated since. And the abbreviation for this, people just talk of ASPR. So ASPR governs everything that's done in animal research in the UK. It's enforced by the Home Secretary, which is one of the highest ministerial positions in the UK government. and it's the enforcement's the responsibility of the Animals in Science Regulation Unit as part of the Home Office. And it regulates any experimental or other scientific procedure applied to any protected animal, and we'll go through a bit of these terms in a minute, that may have the effect of causing that animal pain, suffering, distress, or lasting harm. And this actually includes the breeding of animals for research as well. So just having animals born to be used in animal research, even though being born is a natural process, that is covered under ASPA. And what's really, really important to understand here is that failing to comply with this can be very, very serious indeed. It can result in criminal prosecution. It can result in the suspension of work, not only in your laboratory, but the entire establishment that you're working at. And it can cause severe national and, in cases, international institutional reputational damage. So there are big issues if you don't follow ASPA. But terminology. So I've been saying phrases without actually defining what they mean. So I've said protected animal a few times. So what actually is a protected animal? And this is defined under the Act. So a protected animal is any living vertebrate other than man or any living cephalopod. So any living vertebrate past 2/3 gestation. But then what does it define as living? So an animal is considered to be living until its circulation is stopped permanently or its brain is destroyed. And this is a very, very important definition that is really drummed into our students' heads when they first start trying to do this work, is if an animal has a circulation or its brain is intact, that is a living animal, even if it might be completely unconscious, that is a living animal. You cannot do a regulated procedure on that animal, which we'll talk about in a minute, without breaching ASPA. As soon as the circulation is stopped or its brain is destroyed, that animal is no longer considered living, and you can do what you like to it. Essentially, it's not covered under the Act anymore. So confirming death in the UK before you do anything with tissue collection is very, very important. Finally, what's a regulated procedure? A procedure is regulated if it's carried out on a protected animal for a scientific or an educational purpose, and it may cause an animal pain, suffering, lasting harm, or distress. But this is what it's considered. This is the threshold, okay? So the threshold is deliberately very, very, very low. So pain, suffering, distress, or lasting harm equivalent to or higher than those caused by inserting a hypodermic needle, according to good veterinary practice. So the simple act of putting a needle into a mouse is enough for that to become a regulated procedure, okay? So the threshold is very, very low. So the way we do our animal work in the UK, this is governed by a licensing framework, basically. There are 3 types of license which cover the person doing the procedure, the project where the person's working under, and the establishment, the place where the work's being carried out. So we start off with the establishment license at the top. This is a license given to an institution where the work is carried out, and it must normally be specified as an establishment license. The establishment license holder is usually the director of that institution. It's somebody who has to have the institutional respect and able to enforce and make sure that the rest of ASPA is enforced during the experiments. The project license. So in the UK, if you want to do a body of work on an animal, for example, I want to test the ability of antivenoms to prevent lethality in mice, I need to have a specific project license. During that project license, I can't change my mind and say, I want to see how antivenoms affect blood coagulation, if they do or not. I would have to get a different project license from that. So project license outline a very, very specific body of work. They must be authorized. And within that, it lists the procedures that can be done under that license. And then you have the personal license. So this is a license granted to the person who's carrying out the work. And to get a personal license, it's a lot of effort to get a personal license. You first need to go on a training course and get a certificate. That training course has to have a practical aspect to it. Once you've got your certificate, you then have to apply through somewhere that has an establishment license to get your personal license issued. At that establishment, somebody has to vet you and make sure that you are an appropriate person to do that work. Once you've got your personal license, you can't just start doing animal work. You are only allowed to do procedures specified on a project license or procedure that you'll be doing. So intravenous injection, subcutaneous injection, something like that. First, you have to practice on cadavers. Once you've become competent on cadavers, you're then allowed to practice on live animals under supervision. And only until you've been supervised and then signed off by a competent person, you are deemed to have competence and can do that without any immediate supervision. So there is a lot of layers of supervision here. And then on top of that, you have continual oversight. So you have the animal welfare and ethical review board. So every establishment will have an AWER that continually goes over animal work that's been done or is proposed to be done, and make sure that animals are being treated correctly. You have compliance officers working in the establishments. You have the named veterinary surgeons. So each establishment has a vet on hand at all times to help out with any problems that may come up. And also the Home Office, the government can do unannounced inspections. They can walk in on you mid-experiment and ask you what you're doing to make sure you're being compliant with your license. On top of that, you've got annual reporting. If you don't do your annual reporting, you can get in serious trouble. And throughout your license period as well, you're required to submit reviews. So there's many layers of compliance and oversight. So a project license, it's valid for a maximum of 5 years. And these are the important licenses, in my opinion, because these really govern a body of work. So it's valid for 5 years. Once that 5 years is up, you need to then renew it. To submit a project license, which it's like a very large grant application, basically. So it's a huge document. It needs approval from the Animal Welfare and Ethical Review Board first. It then has to be reviewed by the Home Office, by the inspectors. It's then issued, and then the Home Office can revoke it if the conditions of the license are breached or not met. And what's important about a project license is it enables specific regulated procedures. No two project licenses are the same, okay? And you can only do what is specified on the license, and it is very particular. So if you say you're going to administer a drug intravenously, you cannot then change your mind and give that drug intradermally or any other route. You have to give it intravenously. If you give another route that's not specified on your license, you are in breach of your license and potentially breaking the law, okay? You can amend a license, but it's a very long and arduous process. So you can only do things that is explicitly specified. You can't basically go into the lab one morning and say, I feel like I'm going to do this today. Let's see what happens. And all regulated procedures under a project license are given a severity rating, which cannot be exceeded. Now, we'll talk about severity categories. So there are 4 severity categories in the UK: non-recovery, mild, moderate, and severe. Non-recovery is where a procedure is performed on a mouse where it's under anaesthetic and it will not recover from that anaesthetic. To kill the mouse at the end of the procedure, you will give it an overdose of anaesthetic and it'll die under anaesthetic. So they will not recover consciousness. You then have mild. So mild is the lowest level of suffering that you can inflict on an animal, where animals are likely to experience short-term mild pain, suffering or distress, or it's a procedure where there might not actually be any significant impairment of the mouse's welfare. For example, if you wanted to take a blood sample from a mouse, that would be considered a mild procedure, mild severity limit, okay? A moderate severity limit is where animals are likely to experience short-term moderate pain or long-lasting mild pain. So it's not just the acute pain that's felt here and is governed by the severity ratings, it's the temporal, it's the chronic pain as well. You've got to consider the time frame where this pain or distress is going to be held. Or it could just be that if you're withdrawing water from an animal for a week or so, that would be considered a very severe procedure. Or any procedures that are likely to cause moderate impairment or just the well-being or general condition of the animals. And then you have severe. Now, severe is the severity category myself and Amy work under quite often because envenoming is a very severe event. And this is where animals are likely to experience really severe pain, suffering, distress. It's often where an animal can die from the procedure that's being implemented on it. Or it could be long-lasting, moderate pain, suffering, or distress. or it could be procedures that are likely to cause severe impairment or impairment of the well-being or general condition of the animals. So an example here would be if you are breeding genetically modified mice that have a genetic deficit in them, which causes them to have brittle bone disease. Because those mice are going to suffer broken bones throughout their existence, the birth of those mice is rated as a severe category because of the long-term welfare and distress on the animals. Can I turn this off? Battery on. Turn it off and on again. Okay. No, it should stop working. Okay, well, we'll sort this out. Does anybody have any questions at the moment? No? There we go. Thank you. Now, what's really, really important to consider is that animals should be killed before a severity limit is breached. In project licenses, these severity limits are ceilings that cannot be breached. If you've got an animal under a mild severity limit, you cannot do a procedure on it that will make it go into a moderate severity limit. If you do, you are breaching the terms of your license and you're in big trouble. Like I said, it can have serious ethical and legal complications. It does happen occasionally because of things that if you're working with new drugs, new procedures, things happen that you cannot foresee. So it does happen. So you're not in serious trouble as soon as it happens. But if it does happen, you have to stop what you're doing. You have to report it to the Home Office, and then you have to fill out a load of paperwork to make sure that does not happen again. If it then happens again repeatedly, that's when you can be in severe trouble. So how do we know what severity limit the mice are under and what severity limit they are experiencing at the time? And this is, well, we have to do mouse welfare assessments. So we need to do frequent monitoring. In our case, when we're doing envenoming, it is constant monitoring of animals for signs of pain, suffering, and distress. But also, what's really important is to do routine monitoring of the animals while they're not under a procedure. So it's to check for any health or welfare problems. So in the UK, all mice have to be checked at least once daily for signs of welfare concerns. Because ultimately, if you have a mouse that is not showing normal behavior, it looks a bit sick, you should not be using that mouse in an experimental procedure because it can affect the outcome of your experiment. So this is a rundown of some signs of pain, distress, and poor health in mice. So the first one's appearance. So here we have a mouse which is in quite severe pain. So its appearance is hunched. It's got piloerection, so you can see the hairs on it, the hairs on the mouse are all stood up. It's got sunken eyes. It's not been grooming itself properly. Its face is quite scrunched up. So this is a mouse that is in quite severe pain. The behavior of a mouse. Are the mice inactive? Usually, if you open up a mouse's cage, the mice should be exploring and seeing what's going on. If they're just sat there not interested in their environment, something's clearly wrong with that mouse. If they're writhing about, if they're not walking properly, if they're twitching, all these behavioral traits can show severe pain and distress. These are more chronic ones. Do they have a loss of appetite? Have they got weight loss? This needs to be, weight of mice should be monitored daily. And if they do have weight loss, is it due to illness or a procedure, or is it due to social constraints within the cage? Some male mice do a lot of resource protection, and they will actually prevent other mice or particular mice in a cage from actually accessing food. So you could have weight loss from there as well. Response to stimuli. Are they more aggressive when you handle them? Are they a bit more bitey? Do they then become quiet? Are they unresponsive? Are they segregated from the other mice in the cage? Because mice are social. Another way that can be used, and this has been in trial quite successfully in the UK, is the Grimace scale. Okay, so this has been shown to be very, very effective. So changes in the facial expression profiles of mice, not just mice, you can do it in rabbits and rats as well, can provide a very accurate means of assessing pain in an animal. So it features specific facial action units, so which increase in intensity and response, okay? So at the top, we've got orbital tightening. So you can see this is a healthy mouse here. as the pain increases, the eyes tighten. You've got a nose bulge here. So again, healthy mice looking nice and happy. This one's got a very, very scrunched up face. Cheek bulge, again, it's puffing out its cheeks. Ears back is a classic sign that a mouse is in severe pain. And the whiskers changing as well. So these mice are in severe pain, and these are more in moderate pain here. And you can score these, and they can be used as part of clinical assessment. Okay. These posters these posters are available. They have been developed by the National Centre for Replacement, Refinement and Reduction in Animal Research. They're freely available on their website. So I mentioned the social nest ability of mice as well, appreciating their welfare. So it is very important to analyze a group's behavior as well. Are mice nesting properly? What is the quality of their nests? Are the nests well defined, or are they kind of scruffy and poorly made? Are single mice isolated? So down here in this cage, I'd be a bit worried that these mice are isolated when really mice are social. They tend to like to sleep together. Are they over grooming? Are they fighting? Are they resource protecting? Okay. So with all of these are pretty much useless if you don't spend a lot of time observing normal healthy mice. You have to understand what a normal healthy mouse's behavior is, what they look like, to be able to really accurately assess their welfare. Okay. So now we're going to move on to humane endpoints. Okay. So this is something that is enshrined under ASPR. We have to use humane endpoints. Death as an endpoint in experimental procedures in a lot of places is forbidden. It's not recognized as suitable. In the UK, we are not allowed to do it in most cases. And instead, we have to use something that's known as a humane endpoint. So a humane endpoint is something that describes the identification of clear and predictable and irreversible criteria which substitute for more severe experimental endpoints, usually death. Okay. So you should be able to identify something in your mice before they die, which is a predictor of their death. Okay. So the idea of this is that you can ameliorate or prevent prolonged suffering in a mouse. If you can get your information without having that mouse to suffer on and die essentially through natural causes, if you can ameliorate it during the experiment early, you are then refining that experiment quite substantially. Okay. And these are absolutely essential where severe suffering or death may be involved. So acute toxicology, we use humane endpoints in our envenoming research, animal models of infection, sepsis as well. We use humane endpoints quite a lot. And it's also cancer and neurodegenerative inflammatory diseases. Cancer especially, because if the tumors can get too big, it can be really, really quite distressing for the animals as well. It's very painful. So here are some examples of some humane endpoints. These are more moderate ones. So you could have a body weight drop, for example, 10% in a week. Body temperature if it falls below 34 degrees. These are not set in stone. They will all be specific to the project license, okay? Reduced movement. Doing continuous monitoring of animals now and continuous temperature monitoring of mice is really quite easy and accessible. You can get infrared cameras for smartphones and set up really quite informative systems just using a cheaply available smartphone camera and a smartphone. The University of Newcastle in the UK has pioneered this technology really. So I recommend you look at some of their publications on using smartphones recently. Pathological features, if the mouse develops an abscess, if their tumor gets too big, or if their blood oxygen drops below a certain weight. Okay, so they can be very different. So at CSRI, where we're looking at endpoints, our endpoints are basically immobility. Can the mice, so that's paralysis in the case of neurotoxic venom, so for like a black mamba, or it could be loss of self-righting reflex. So if you turn a mouse over and let go of it, it should immediately stand straight back up. If they can no longer do that, that would suggest that mouse is at an endpoint. Seizures, external hemorrhage, or labored breathing with dyspnea, they're all common envenoming endpoints. Under a license, if a mouse reaches one of these humane endpoints, it must be killed to end suffering or risk a severity breach. And then that comes on to killing, okay? So ASPR also explicitly sets out methods of humane killing. And these are called Schedule 1 because under ASPR, there is an annex, essentially, which is called Schedule 1, and it lists how you are allowed to humanely kill an animal. The methods under Schedule 1 are animal and size or life stage specific, okay? So there are lots of different methods on there, but you cannot use them for all the animals or all the life stages. For mice, typically, what we're allowed to do under Schedule 1 is dislocation of the neck, exposure to carbon dioxide in a rising concentration. It must be rising. You can't just put them in 2% CO2. It needs to be a lower level than brought up, or an overdose of anesthetic. It can only be done by a competent individual. Okay, so this needs to be an individual that has been trained by somebody who else is competent. On a lot of these, you will start on cadavers. And the reason for this is killing an animal is quite a distressing procedure for the person as well, and it needs to be done correctly. If there's any sort of hesitation during the procedure of humane killing, you can actually increase the suffering and the distress of the animal, which is not what you want to do. Non-Schedule 1 methods, so using chloroform, ether, exsanguination, are expressly forbidden. You cannot do them unless they are explicitly permitted under your project license, okay? And if you do a Schedule 1 that is not permitted on your project license, you can get in serious, serious trouble, okay? So, there's only 2 links there for resources on welfare, but they are absolutely excellent and very, very comprehensive. You have a lot of sort of crib sheets and cheat sheets and posters that you can go through. I really highly recommend you look at these because they have a lot of free accessible material, okay? So that's my overview of legislation and mouse welfare. I'm now going to hand over to Dr. Marriott, who's going to talk about more of the practical implication of these and in improving mouse and animal welfare, not just mice, dogs, and cows as well, in NTD research. Thank you very much.
Okay, so thanks for inviting me here. So I'll follow on with sort of more examples of how we've sort of improved in vivo models within neglected tropical disease research. So just a bit more of a background on me. So my sort of expertise lies within developing new in vivo models which minimize animal usage, refine animal usage, also explore the use of looking at ex vivo and in vitro models which better replicate what's going on in vivo to, again, by that way, reduce animals used for testing. And also a lot of my research has been very much drug development focused, especially on the in vivo side. So sort of developing the models and any sort of drug candidates coming through, using these models for efficacy testing and pharmacokinetic testing, profiling and modeling. So that's sort of all I do in a little nutshell there. So to start with, lymphatic filariasis is which what I've spent most of my career working on up until recently. Hopefully some of you know, but then, yeah, so you've got lymphatic filariasis, which obviously is a parasite transmitted by mosquitoes, which is in the lymphatic system and can cause edema and other problems. There's also a subcutaneous form, onchocerciasis, also known as African river blindness, which also is spread by an insect vector and can cause lots of problems. There's also loiasis, so the eye worm. So this wasn't considered an actual sort of issue until sort of the Mass Drug Administration program started for lymphatic filariasis and onchocerciasis, where sort of it would, in patients with a really high sort of parasitemia, would cause severe adverse drug effects. So as mentioned with the Mass Drug Administration programs, these target only the sort of transmissible stage of the disease, so what the mosquito is transmitting. So once you have an established infection, that's really hard to treat. And also, you know, you've got issues with the severe adverse reactions if you've got co-infections with loiasis, onchocerciasis for the treatment she's there. So currently, you know, sort of what we wanted to do was target the Wolbachia, which is a bacterium inside the worm, so helpful for growth and development. So doxycycline antibiotic can sort of deplete the Wolbachia inside the worm, which causes a slow death. But this is a very long treatment regime, so about 8 weeks. and can't be used in pregnant women or children under 8. So the sort of aim of what we wanted to do for this project was to look at new specific therapies, specifically targeting the Wolbachia, with a shorter treatment course, with retaining a high safety profile in those co-infections with loiasis and onchocerciasis. So the sort of problem that came about was there's no in vitro model of the adult stage parasite, so the parasite stage that we really want to target. There's also no small rodent model of loiasis. A lot of it was based on baboon models, so a heavy reliance on non-human primates. There is a natural gerbil model of lymphatic filariasis. They're a natural host of the parasite, but as they have a competent immune system, the level of variations is really high. So that if we wanted to use that for an experimental model, sometimes you'd get sort of parasite failures. So you'd have to sort of compensate by that by using more animals, which obviously isn't the most ethical or cost effective. So basically what I'm going to go through is sort of the pipeline and how we've developed models and to reduce animals based on this drug discovery pipeline. So more when, so developing the model, then when sort of compounds or drug candidates have been identified from in vitro screens, further developing these models and taking them in vivo to get a proper sort of drug profile, know what's going on with the toxicity before we even put them in an animal model. So that all hopefully should cover in this sort of session. So again, as Stu's mentioned, everything's based around sort of the 3 R's. So minimizing animal usage. If you've got an in vitro or an ex vivo model that you could use before, you should always absolutely use that to get an idea before you progress something in vivo, and always do the best to seek like replacement models. So even like lower animals, so rodents rather than dogs or monkeys, and just basically like be more mindful of what we're doing and have a heavy justification for why things would need to go into an in vivo model. So go through sort of what we've done for this in vivo model development is the sort of whether the rodent model you use, is it an appropriate species? What sort of how many numbers of parasites can you get back? And sort of maximizing the scientific outputs with the minimization of animal studies. So once sort of this was established, we wanted to look at PK profiles of any like lead candidates and model these for different scenarios to really like sort of inform the dose regime to put in mice, so everything sort of translates back to the human scenario basically, and just validate everything before sort of moving forward and making too many assumptions. So what I sort of done is develop this model, also developed an adult stage parasite ex vivo model. So that's a parasite of interest that's a drug target. So there's these in vitro models which look specifically at the Wolbachia, but obviously if you've got a drug targeting that, it might not work as well against the whole actual adult parasite. And then again, it might not work in an in vivo system. So what we did to try and develop this ex vivo model was sort of link it back to sort of the quality of the parasites we were getting from the mouse model, to make sure that sort of any drugs we tested in this adult model would be representative of what's going on in vivo, so would be predictive. So we'd be able to test sort of compounds at different doses in this ex vivo system to give us an idea of what's going on and a sort of go, no go. So if something didn't look that good, we wouldn't progress it to a mouse model. So that's sort of reducing animals that way by putting in a sort of another bridge gap there. And sort of once we've done that with any candidates, that sort of links back, so everything sort of ties in together. We then look at the pharmacokinetic profiles and then put that together to do pharmacokinetic, pharmacodynamic efficacy testing of the lead drug candidates. So once we've got all that information from the new models we've developed, we'd then be able to test properly and be really certain that something would work before it goes in an animal. So for the model selection and development, so as I mentioned, the gerbil is a natural host of filariasis. But when sort of some drug studies were being done, there was sort of a lack of translation between gerbil models and then a sort of a dog model, with you having to, through the preclinical testing profile, testing two species. So what we sort of wanted to look at was different immunodeficient mouse strains. So using a sort of immune knockout system to sort of produce more parasites essentially, and look at the variation of how many parasites we'd get back so we could accurately power experiments. So that's what we did there. So what we did to start with, so there's this publication looking at lymphatic filariasis and onchocerciasis models in different immune knockout animals. So what was done here was use sort of skid mice, so very immunodeficient, immune impaired animals, which allowed for a greater parasite recovery compared to wild type mice. So we did manage to do this in these models for lymphatic filariasis, and then applied these to the onchocerciasis parasite as well, just so, because we want to test the drugs, you know, against the cross testing for safety, is ideally use the same species, the same immunodeficient mouse lines, so everything's sort of consistent, if that makes sense. So what sort of did here as well was, so you've already got your drugs in your mass drug administration profiles, is then test those in the mouse model to sort of validate the system to ensure you are seeing drug effects, because if you wouldn't be seeing any drug effects, there's clearly a problem with that model, and that's obviously not very translational to the human situation, sort of rendering it very ineffective and kind of a waste of animals. So that was sort of the start of developing the skid mouse model for lymphatic filariasis and onchocerciasis. So again, with needing to maintain this safety profile, especially against lower microfilariemias, that's your blood circulating parasite, is you also wanted to develop this again as a model in the skid mouse. So that's sort of been done here with different immunodeficient strains as well, just to see sort of what's the best. So this was used for like a model development for drug studies essentially, but so in the sort of team, there's like more pharmacology model development based. And there's also, by using these immunodeficient strains, you can also look at the bigger sort of detailed picture at what's going on with the immune response and the host immune response. So sort of by developing a model like this, it allows you to sort of get more data from it essentially, if that makes sense. So this was tested looking at this, these graphs will just show the different parasite recoveries with the different mouse strains, like adult parasites, and the microfilariemic mouse model, which is injected intravenously just to check you can get those parasitemias, so you could do long-term drug testing to compare the safety profiles with your drug in these different models of filariasis. So that was done there. So what sort of we did then to try and, so you've got your sort of immunodeficient mouse lines, which ones work best for parasite recoveries in a drug screening model. But what we also wanted to do was sort of have mice as essentially worm stocks where we could take parasites, you know, to test against in vitro to develop the ex vivo models at the same time. So what we wanted to look at is sort of the variation in parasite load between gerbils and these two immune knockout mice, and look over time. So if we sort of left the parasites in the mice and couldn't use them for 6 months, do you still get a good parasite levels, or is there something going on? So you can see here, you see there's still quite a bit of variation in the mouse models, but the parasite load is a lot higher than the gerbils. So hopefully that gets the picture across that using mice is better, because if you've got gerbils and you've got a lot of sort of infection failure rates, you'd have to compensate for that, especially in a sort of drug screening scenario when you want the parasites back. You need enough to be able to do your testing to power experiments. So it all like interplays with that and looks at a detailed picture here. So this is sort of utilized. So there's not a massive difference between using the skid mice and this IL-4, IL-5 receptor knockouts. But what we found was when we were leaving mice for long periods of time, the skid mice actually declined in welfare. So essentially what we'd use would be the IL-4, IL-5 parasites, strains, as the mice had a better sort of health and welfare, which was important in this system. But what we did as well as part of it was just cross-check the parasites, that everything's the same for all the viability. So this is what we sort of did to get parasites to try and develop an ex vivo model. Gone too fast. So what we did here was try and sort of replicate the lymphatic system in a sort of culture dish. So we've got lymphatic cells and some sort of naive and stimulated macrophages. So what we did was leave these in culture, replenish the media, and also use a mouse model to compare the parasites at the endpoint. So you can see on the graph on the left that the parasites survive quite a long time in vitro. So we looked at different time points along that to check the health and fitness of the parasites to make sure, again, that this ex vivo model would be representative of an in vivo model, so to sort of reduce and refine animal usage that way. But when we looked at different, as you can see in these other graphs, at different time points along the culture course, they actually declined. So the levels of Wolbachia were slightly different and the metabolic activity was slightly different. So this was a bit of an issue. So we decided to leave sort of the parasite culture system as 14 days. Obviously, we would want the Wolbachia levels, as that's our drug target, to represent those in vivo. Otherwise, we'd get false readings in this in vitro model, which would then cause us to put drugs into the mouse model, which would never have worked, essentially. So just by having this, it can further screen out drugs to reduce and refine animal usage. And then again, just validated this as a drug screening model in vitro, comparing sort of the levels of Wolbachia depletion against what you'd see in vivo, again, just for like robustness to check everything's sort of representative of everything. So then sort of once these models are developed, the traditional sort of pipeline is to look through sort of the pharmacokinetic properties, which you traditionally do in your sort of species of interest and strain of interest without having your infection present. So you want to really be looking at like how much of your drug's absorbed, how it's distributed, metabolized, excreted, and its toxicity levels. Because, you know, something might work really well in vitro when it's a constant exposure, but when it actually gets into the body, it might not be able to be absorbed, and that can be a problem. So you sort of want to look at this before doing the pharmacokinetics in a mouse, because again, to screen out sort of what might work and what might never work. So it would be a waste of animals putting them straight into a mouse. Sort of what we can do to look at absorption is to look at these cacao 2 cell layers, where you've put the drug in one section and sort of just measure how much drug comes out the other side. You can also do metabolic studies in vitro, so looking at liver, hepatocytes, and microsomes, which have got their metabolizing enzymes, which is what you can do in vitro, and do in vitro ex vivo toxicity testing to see sort of any toxic effects of the drug. For distribution and elimination studies, obviously, it's very difficult to do in vitro. So what you'd do would be to label a drug, either radio label or fluorescently label, administer it to your animal and sort of see over different time courses where the drug builds up and where it's eliminated. So you can get an idea of that, in addition to getting an idea of then drug exposure. So as we can't do this in vitro very easily, so in vivo is what we still try and incorporate is what Stuart mentioned before, is refining this, so using microsampling for your bioanalytics when you look at this down the line. So instead of using, say, 1 mouse per time point per treatment, we sort of optimize these bioanalysis protocols so that we just take micro blood samples to be able to do our analysis on that. So again, then you could sample from the same animal multiple amount of times as it's still within your sort of project license to take that blood volume. So you're sort of, again, maximizing the data you can get from the mouse while minimizing how many you're using, if that makes sense. So then sort of when we put it into the mouse model, essentially when we know what sort of drugs look good and have got these sort of best drug-like properties and pharmacokinetic properties, is so you'd administer it and then you sort of build these profiles to see what's going on. Are you reaching your sort of therapeutic level, not reaching toxic levels, which is where you'd start looking at toxicity testing here as well. And then you'd start using sort of modeling software to play around with the doses to reach that sort of exposure level that you want. So when you sort of got all this together, like again, building in your in vitro and sort of your refined sort of animal models to do this, you can then use, put everything together basically to use as efficacy testing. So it just shows sort of how sort of the SCID mouse model has been used for sort of your bruiser, so your lymphatic filariasis parasite, and then shown to be optimized for your sort of cross-testing and safety testing with onchocerciasis and loiasis. And then also we've done the PK testing and profiling to see sort of what drug exposures are reached, and what can be done and what is done here is say if you're using a repurposed drug, so one that's already used for something else, and applying it to a new disease, is you can take the sort of dose that would work in a human and replicate that in a mouse to get the exposure. So say you want to reach a sort of exposure of something which is done in a human with one dose, you may need to dose twice in a mouse model to reach the same exposure, and vice versa. So just that helps, with the translational aspect of the model, to help sort of progress your sort of data and get the most out of the model. So sort of outcome of all this sort of pipeline has managed to develop a immunodeficient mouse model, which can replace high mammals such as dogs and primates, and sort of use this to look in more detail at these different immune deficient strains, to look at parasite production and the variations, so you can really design the studies appropriately to make sure you're not using excess mice. It's all based on scientific justification for how much, how many mice you should be using to get your sort of significant differences in what you need for your statistics. So sort of as a result of all this, which has been through the Anti-Wolbachia Consortium, so drug discovery program, is through using these models and pharmacokinetics and pharmacokinetic modeling for human exposures as well, is this has led to 2 compounds currently being in clinical trials for filariasis, which is really good, and just shows as well how using these and developing new mouse models and refining animal use is still really given good results, where you've got 2 compounds that have now progressed to clinical trials. So as another example, which is very similar, is there's also veterinary filariasis, which is sort of in the same sort of clade as your subcutaneous parasites, so loiasis and onchocerciasis. So this can be quite harmful in dogs and cats. It's a big problem in America and now in parts of Europe as well, again spread by mosquitoes. So the sort of way this is treated prophylactically with macrocyclic lactones, which target the sort of larval stages. So basically, if a dog's bitten by a mosquito and gets this parasite in its sort of bloodstream and it develops, that drug would target that then. However, if for whatever reason a treatment's missed, it's expensive, it's monthly, people forget, and just not responsible pet owners, if it establishes an adult infection which can be in the heart and be fatal, there's not actually that many treatments. There's only one called melarsomin, which is curative, but it can cause severe and fatal adverse reactions. It's also a really complicated treatment course, so the dogs would need to be sort of pre-treated with steroids, not allowed to exercise, so very restricted and very difficult, and can still be fatal. There's also no treatment available for cats. So sort of based on our sort of experience with the human filariasis, was to use the same sort of approaches by developing new therapies to target the Wolbachia inside the parasite to get a slow death, so it'd be safe, which would sort of get rid of problems with resistance, which is being seen with the prophylactic treatment, and also sort of get rid of any sort of fatal adverse reactions with the current adult treatment. So the sort of problem with this, though, was that there's no small animal model available. So any testing for any new therapeutics or vaccines is done in dogs. So it probably sounds a bit odd, as dogs are the sort of, subject to this infection. But for any sort of experimental testing, it's still using a lot of dogs. Again, you get some variation in parasite loads. So for any experimental procedures, dogs would actually be infected with a massive sort of bolus of parasites, much more than they'd be exposed to in the wild. So obviously that's not very good. We want to move away from doing sort of larger animal testing. So you can just see in these graphs how many sort of dogs and cats were used for these purposes previously. So what we wanted to do was sort of use our knowledge with developing mouse models for filariasis and lower to apply these and see if you could develop a model of heartworm here to replace the use for dogs for this initial testing, before you know that something's going to work. And also, again, look at in vitro testing the same way as we did with the human filariasis. So what we did was we sort of selected the mouse models based on previous data, what we'd done, and our knowledge of what we'd done for human filariasis mouse models. So we had an idea of what may work based on the sort of immunology. We also used some steroids based on previous parasite work with a different parasite in a mouse model, which seemingly bolstered parasite loads. So what we saw is we get quite good recoveries when we use these strains for a shorter larval time course period. So you'd get back about like 5 to 10% parasite retrieval, which is sort of comparable to what we'd see with human filariasis. We also wanted to sort of check where the parasites were going to make sure that would be sort of in a similar region to what would be going on in the actual dogs. And by this stage in the infection, we showed that they were staying in the skin and subcutaneous tissues, with some going to the muscles, which is what you'd expect looking back at the literature and meta-analyses of what was found in experimental dog models. So again, as we wanted to use this as a screening tool for anti-wolbachioal therapies, we also had to look at the sort of whether the parasites were growing consistently and whether the sort of Wolbachia levels were also expanding. Again, this is based on our knowledge of what we've done for human filariasis. So then what we did as well was look further in detail on this, and what we did to sort of further develop and validate the model was also test this at a different laboratory, so one in America, with a different isolate, just to see, whether it would be applicable for in different situations and with a different parasite strain, so like resistant strains as well, which we found you did get good recoveries. So it was a good basis for a model. We also tried to do in vitro sort of model development, again, as the replacement. However, the parasites, even though they survived like a little bit in vitro, they were just not comparable to the sort of growth levels in vivo. So we just sort of didn't do this, as it would just be sort of pointless because you wouldn't get any usable data that was translational to the mouse model. So what we sort of did as a solution was grow the parasites up in the mice for a few days, then take the parasites out of the mice and do drug testing ex vivo there, because we found that was more stable. That probably sounds like an odd way to do things, but that is still considered a refinement, because you're taken away from needing to actually subject the mice to more procedures such as dosing. So what we do is do this and get an idea of whether any drugs are working, which give you better justifications, then go back in vivo and do this properly. So that is a refinement, and sort of again using as a screening tool to reduce sort of any compounds that wouldn't work. So again, essentially can reduce animals that way. So when we did this, we looked at the drug responses, so validated it using sort of typical drugs that would be used against this stage to generate this data set, and also validated the in vivo model as a drug test using the gold standard therapies, so those macrocyclic lactones, where we'd see a reduction in parasite load, which is what we'd expect in the dogs, to just to further justify that what we're seeing would be representative and translational to the actual sort of target model. We also then, as we wanted to use this as a anti-wolbachioal sort of model, as that sort of novel aspect for our sort of drug portfolios, is test these using sort of a reference drug, so doxycycline, and then some of our newer compounds where we're actually able to see a better sort of efficacy. So you could not just look at whether something works or not, you can actually distinguish between sort of the levels of how well different compounds are working. So we're quite happy at this point that we'd validated this as a model to do drug testing in, which is currently under review, so should be hopefully published soon. So sort of outcome of this is reducing the number of dogs used in experimental procedures specifically for this sort of drug testing. And through this as well, we've identified some compounds and are going through the process of testing our sort of human filariasis backup compound series, which have been allocated for veterinary use in this model. So some look actually really good and have progressed, further by using this mouse model. We know look really good to a first sort of canine model of disease. So the second sort of species in your target target model. We've also used this sort of model by sort of presenting at conferences and stuff to gain interest. So other sort of small biotech companies have sort of used this model to then test their compounds. So yeah, so I think that's it. Oh no, it's not everything. That's everything for filariasis. But then just to go again to another neglected tropical disease is just looking at the snakebite model. So basically for everything, all the research done in filariasis mouse model development was to really like maximize sort of, the outputs and just double check that everything is, really validated and as a drug model, and sort of make sure that everything's translational to anything, you'd use eventually in human studies. So just as an example of, a stark contrast essentially is a snakebite model for sort of antivenom development and regulation. So what's done by and is endorsed by the WHO for any sort of approval for new antivenom is pre-incubate the venom or any drug, antivenom or drug with the venom for 30 minutes at 37 degrees. So essentially you're having an effect sort of in vitro before you then put it into a mouse, so intravenously. So it's important to note that this venom dose is like a super lethal dose. So you want to induce death, really. So these are typically monitored for 6 hours and the outcome's live or dead. So again, comparing to other models of neglected tropical diseases, it's not the most, useful for getting a lot of data back to inform, what's sort of going on with your compound of interest. So what's been done, I think, with trying to improve animal welfare is put in these humane endpoints, as Stuart mentioned. So not just leaving the animals to die, but putting in these endpoints when they show seizure, paralysis, and loss of rising reflex, and any signs of external hemorrhage, and sort of build this data set. So this is typically what a sort of snakebite model would sort of look like for testing. So you'd sort of establish an LD50 dose. So you'd test different doses of venom and basically just see at what stage like 50% of the mice die within a time course. And then for your actual efficacy studies, this is then multiplied like 2.5 or 5 times this lethal dose. So again, it's such a high dose to get this like full death group. Your then treatment is using different doses, pre-mixed and put into the mouse. And then it's the sort of live dead at the end that we see as a marker for sort of what looks good and what could be potentially then approved for clinical use. So it's a very like crude model and a sort of worst case scenario, but then best case if your antivenom protects. But again, just to look at the differences between a sort of human snakebite situation and a mouse model, it's very, very different. So you might receive a single like intravenous dose of a very high amount of venom. So they rapidly succumb to this and are very, very sick. It's also not pathology specific. So there's no, you've got different types of snakes that cause different pathologies. This isn't, explored in the mouse model. It's just this live or dead phenotype. Whereas with the human snakebite, your venom is typically injected subcutaneously. So instead of seeing this massive spike in venom dose in the bloodstream, it'd be a slower sort of depot effect where the venom would be leaching out. So obviously someone doesn't die immediately after being bitten, whereas sometimes the mice can die minutes after venom administration. So it's just important to notice these sort of differences and also how severe the model is. So I'm sort of just showing that it is so different and sort of developing any new models, you want to sort of make it as representative of the disease and the actual sort of disease model and situation as possible. So what sort of come from these studies is actual suboptimal antivenoms can make it to market, which has been highlighted in these publications, which obviously is very dangerous and sort of not, sort of suggests that if we can improve on models to get better data, that would be sort of beneficial to everybody. So what we also want to do in terms of animal welfare is develop a better model that's better reflective of a human snakebite scenario and also minimize animal suffering. So potentially not using such a high dose of venom and looking for different markers during the course of envenoming So looking at coagulation and different immune markers to see if there's something that we can see without the mouse getting so sick. So what we sort of purchased within CSRI are these coagulopathy devices. So this one specifically, the ROTEM, looks at the phenotypic properties of a clot, which they, with any sort of hemotoxic venom. So it uses a pin and rotation to measure sort of the clot formation time and the maximum clot firmness. So this is, and the degradation of the clot. So this is sort of a typical profile of what you'd expect a normal sort of clot to look like. So what we did was test different doses of venom. So we looked at the lethal dose and just sort of titrated them down to see what the effects are, to see, what's going on with the coagulation and if we can potentially, develop a new improved model that would minimize animal suffering by using these as markers. So we looked at different time points just during the acute phase for now, and just to see, what the blood looked like essentially. So this is your normal mouse clotting profile. This is after an hour of envenoming. So this mouse just received placebo, so just PBS. So that's your normal clotting profile. So you've got a nice big clot, your blood's clotting properly. When we injected 1 microgram of venom, so that's a lot lower than what would be used in a sort of efficacy study. You can see that a clot just isn't forming. So you're just not seeing a proper clot formation. It's worth noting the mice were absolutely fine. They showed no signs of any envenoming or any sort of welfare issues at this point. Whereas obviously when we go to the really high doses, they're very, very sick and quickly reach those humane endpoints. So we found this really, really interesting, just to show with just such a lower dose, how much of an effect you can observe without the mouse needing to be so ill, basically. And when we increase the dose, that's basically just showing that there's no clot formation at all. So even with lower dosage, you get no clots forming. And with the higher dose, because the blood was sort of that much of a mess, we couldn't get enough back to actually run these studies, which I think just goes to show, just how much of an, using such a high dose when we could potentially be using lower doses. And sort of using markers to make it so more relatable to a human situation, and give us an idea of, if we go into testing antivenoms and drugs, what sort of get more data, what's going on kind of thing. So you can see as well when we do a time course of this 1 microgram of venom, after 15 minutes of envenomation, there's no clot formation at all, and you can see a slight recovery at 30 minutes and 60 minutes, which is something we want to explore further, and also look at different doses too. And sort of the outcome of this is very, very pilot work. Is just to show that there's evidence of that we can use these sort of coagulation markers as early signs of envenoming at these low doses without causing any harm or sort of welfare or issues to the mice, which sort of gives us a good basis to start developing a new model to get sort of a better outcome, better scientific outputs, and also better for the mice, so we're not using such severe models. And that's just my acknowledgement. So CSRI was all the snake venom work. Anything filariasis or veterinary heartworm was done in Professor Joe Turner's lab at LSTM. So he leads that and the drug development programs. And the onchocerciasis and lower work was conducted out in Cameroon with a collaborator. So I'll hand back to Stu.
Thanks very much, Amy. So we're just going to finish up now with, I'm just going to take a step back from mouse work, and we're going to look more at what the mouse work and the current model that's used for envenoming and testing antivenoms, what that actually means for human treatment of snakebite in sub-Saharan Africa specifically, but also globally. And one thing I wanted to mention as well is, and we've not actually mentioned this throughout the stuff, we're talking about the welfare of the mice and talking about the distress of the mice. When we're doing these really severe procedures on the mice using the venom, we're injecting venom to these mice intravenously directly. It can have a severe impact on the people doing the experiments as well. It is not a pleasant time or a pleasant job to be doing. And really, as much for the mice, it's as much for us, so we don't have to do this anymore. So yeah, discussing the current, make sure I've got this the right way around, the current state of preclinical antivenom testing in sub-Saharan Africa. And I know this is something that is very close to George's heart. So we'll start off. What is antivenom? So antivenom is therapy that's used to treat envenoming if you get bitten. So the way you make antivenom is you collect a snake's venom, which they do here at Case-Rick. You then take that crude venom and you inject it into an animal. Usually it's a horse. 99% of the time it's a horse. Occasionally it's a sheep. And then you would hyperimmunize that animal. So it generates lots and lots of antitoxin antibodies in its bloodstream. And then you take the blood from that animal, you bleed them, you scoop off the red blood cells and the white blood cells, and you put that back into the animal because you're only interested in the antibodies. You extract, purify the antibodies, you refine it, and that is it. It's ready to be used. It's a very, very simple method of production. And the premise is that the antitoxin antibodies within the antivenom will bind and neutralize venom toxins when administered. And it's been that way pretty much since it was developed in the 1880s, initially in pigeons. I'm not sure why it was done in pigeons originally. The way it's been manufactured has not changed massively at all since then. It's still basically the same therapy. And that's a problem because the status quo has basically existed since then, because antivenom is such an old medicine. It's not kept up with new regulation. And as I said, antivenoms today are made in the same way. And this includes, Vipretab in the UK. This was Favafreak before it was discontinued by Sanofi, and Crofab in America. So this begs the question, how are antivenoms approved for human use? So this is a normal drug discovery and approval pipeline, okay? You have your basic research, you then do your drug discovery, so this is technically in vitro, you then have preclinical, which is what we've been talking about in mice. And here, all these white lines are basically different compounds, different drugs that are going through the drug pipeline. You can see as it travels along the pipeline, most of these drugs fall off and vanish because they're not effective. They don't translate. You then go into human clinical trials, you phase 1 for safety, tens of people, phase 2, hundreds of people, and then phase 3, which is your thousands. And then once you've got past that, you have basically your regulatory approval. And this is taken a lot of work. And then after your regulatory approval, we have a phase 4 clinical trial where there is continual monitoring of efficacy, essentially. So surely it's the same for antivenoms. Surely they're a drug used to treat envenoming. Surely it's the same. The short answer is no, it's not. This is a quote from the WHO. Antivenoms are unusual among pharmaceutical agents in that they have been used in human patients since 1896, with little attention being paid to their clinical trials or effectiveness of their efficacy, okay? So far, the vast majority of antivenoms have been registered for clinical use in humans without any demonstration of their efficacy in humans beforehand. And what we do is how it's approved is you use the preclinical mouse model. If it can save a mouse, that very, very artificial experiment that Amy's just described using a very high lethal dose of venom, if you can save a mouse with that, it's good to go into a person. And that just doesn't seem right, especially in the 21st century. So this is the same absolutely everywhere in the world. So Vipretab is an antivenom that would be used in the UK because we do have a venomous snake in the UK, is the adder, Viproberus. It can be fatal. It hasn't been a fatality since the 70s, but there is an antivenom available. It's called Vipretab, which would be given on the NHS if you were bitten. Although it's not approved or marketed, it's available in Europe on a named patient basis, okay? So it's basically available even though there's no clinical demonstration to say that it's effective. So this is ever so slightly outdated now. So if we look at the sub-Saharan African context, here are, as of a few years ago, all the available antivenoms in sub-Saharan Africa. I think there's 18 there. These are the antivenoms available, so it's moved over with clinical studies, okay? And these are of hugely varying degrees. of quality. Some of them are absolutely awful. So you can see maybe half of them have got some demonstration of use in a clinical human study. If we then go forward to clinical trials, only 2 have been used in clinical trials. And that's Equitab G and Equitab Plus, which was used in Nigeria in the noise. Equitab G is a monovalent antivenom. It's only against Echis. Equitab Plus is a trivalent. It's against Echis, Bitis, and Nausea nigricolous, the spitting cobra. But the clinical trial wasn't against all 3. It was only against Echis. So it's had a clinical demonstration, very robust clinical trial, only against a single species. And then there's this fantastic review that I highly recommend you guys go and read by Julien Potet at MSF. Some products are available in Africa commercially, although they've been found to lack effectiveness. So even though it's known they don't work very well, you can still use, they can still be procured and they can still be used to treat people. And this is obviously a serious problem. So how are antivenoms marketed? So basically, these are 3 boxes of antivenoms that are available in sub-Saharan Africa. This is Ainsworth, which has recently been recalled in Kenya. This is a product from India. This is Vin's Central Africa, which I'm hoping has been discontinued. Been told it has. And this is Vin's, not Vin's. This is Premium Serum's Pan Africa product, which has been approved for use in Kenya. And basically, the way they are marketed, if you look on the side of the box, I appreciate you might not be able to read this. They've basically got these claims on the side of them. So this product here says it can, each mil upon reconstitution neutralizes not less than 0.45 milligrams of dried black mamba venom or 0.6 milligrams of gaboon bag venom. Or this here, it says this neutralizes no less than 25 LD50s of puff adder venom. Now, if you were a clinician looking at that, you would read it and say, oh, this can neutralize 25 LD50s. That sounds like a big number. That's pretty good. What they don't say anywhere on these boxes is that is a mouse LD50. So that neutralizes 25 times the amount to kill half the mice in an experiment. Here as well, 0.6 milligrams of Bitis gabonica venom. If you know anything about Bitis gabonica, 0.6 milligrams of venom from a single bite is not a big amount of venom. So without context, this is absolutely useless, okay? And unfortunately, many low middle income countries, they don't have the ability to verify manufacturers' claims against local venoms, and that's what's really important. They don't specify what country their venoms are from, and people know venom variation is a massive issue. A puff adder in Nigeria is not the same as a puff adder in Kenya. So basically, what purchasing departments in ministries of health have to do is basically take manufacturers' word for it. And this can lead to serious problems. So this was sort of a watershed paper where, so this was published in 2008, failure of the new antivenom to treat Echis ocellatus snakebite in rural Ghana and the importance of quality surveillance. So essentially what was happening here is they were using Fabafrique in Ghana, which was a very good antivenom for treating Echis ocellatus, but it's extremely expensive. An Indian company came along and said, we've got an antivenom that does exactly the same as Fabafrique, but we can sell you it for 10% of the price. So obviously they switched because you could get 10 times more vials of antivenom. Problem was when they actually used this antivenom commercially, the mortality rate went up from basically 1.8% with Fabafrique up to 12.1%. So the 10% more people were dying when they switched this antivenom. So it's really important. And this would have been stopped if there was a quality surveillance in place, okay? And that's not the only case. There's also, this is the product that was available in Kenya. I think, George, you bought this vial in a pharmacy in Kenya. Oh, hello. What's in the name here? Why is it? I need to go back. Here we go. So this is a product specifically marketed for Central Africa, and it says it's effective against black mamba, Vitis carbonica, and then these two snakes here, Deboia russellii, Russell's viper. You do not get Russell's vipers in Africa. You get them in India and Far East. Echis carinatus, saw-scale viper. You do not get Echis carinatus in Africa. Echis carinatus is found in India, in Pakistan. Echis carinatus in Africa, well, the Echis species within Africa used to be known as Echis carinatus in like the 70s, and then the terminology changed. So this antivenom here has been made with completely inappropriate antivenoms, utterly useless, but was on sale, marketed as an African antivenom. So there's serious, serious problems. And when it comes to it as well, so for example, this Echis carinatus venom, so this is Echis carinatus from the UAE, and we've got Echis carinatus from India. The venom composition between the same species, so these pie charts show the venom composition of the different toxins, even within the same species, they're massively different. You move into the different species that inhabit sub-Saharan Africa, so Pyramidium leakii in Kenya, they're completely different. So this antivenom would be very, very poor at neutralizing the bites of a Kenyan Echis. So what are these claims made on? How are these therapies licensed? And it is based on that artificial murine preclinical lethality study. All the claims are made on mouse data alone. So why do we not have normal clinical trials for antivenom? Why do you not have that human clinical efficacy beforehand? And there's a few issues for this. One is there's a lack of regulatory oversight, and there's a lack of interest predominantly in snakebite research up until more recently. The second thing is venoming is a medical emergency. It needs to be treated with there and then. There is a good long history of antivenoms being used and being effective. So it's got that on its side. Clinical trials are difficult because you can't really have placebo groups. It is unethical to have somebody present with a snake bite and not treat them and give them something that will not save their life. And for a lot of snakes, there's simply not enough bites in enough areas to perform clinical trials for some by envenomation from some species. It's just not possible. So all that okay. But that doesn't dismiss the fact that clinical trials have and can be performed. And what Dr. George is hoping to do and what we're hoping to do at CSRI and lots of collaborators around Africa and indeed around the world is to make clinical trials more norm in the future. And what I want to note from that, and what I find really, really perverse, is it's not the antivenom manufacturing companies that are doing the clinical trials. It's academics doing it on behalf, not even on behalf of doing it because the antivenom manufacturing companies simply don't do it because they're not required to do it. So they'll lose money if they do it. So but if it was any other pharmaceutical, it would be, the onus would be on the manufacturing company, the pharmaceutical company, to perform those clinical trials. But baby steps. So as Amy's been through this, neutralization of lethality, the effective dose 50. And you get this readout of basically mgs of venom neutralized per mil of antivenom, mg per mil, okay? It's not a very effective demonstration of an antivenom's effectiveness in a human because it's a completely artificial model. The pre-incubation, the direct intravenous injection, the pathology of death that you get in the mice is not reflective of how humans get bitten, humans develop their pathology, and ultimately humans die. However, basically with your ED50, if you get a high ED50, you get an antivenom with a high potency. So you basically want antivenoms with good ED50s. You want them to be able to neutralize as much venom as possible per milliliter. If your ED50s are starting to get on the low side, antivenom is going to be completely useless. The problem is there is no defined cutoff for what an effective ED50 is, okay? And at the moment, my personal opinion is that some companies are gaming the system in that if you can demonstrate that you can induce survival in this model at a really, really, whatever dose of whatever dose of antivenom, even if it's completely ridiculously high, so you get a low LD50, that is still suitable for use in a human. So just because it can provide protection in this model does not mean it will provide protection in a human, but it is marketed in that manner. So what's been done about this? So since Snakebite became a WHO NTD, made it on the NTD list, WHO has taken a few steps, and you've got to remember the WHO is severely underfunded. They don't really have a lot of money to do a lot of things, so they have to be very, very targeted with what they're doing. But one of the things they are putting a lot of effort into is this WHO antivenom pre-qualification scheme. So basically what they're doing is they are inviting antivenom manufacturers to submit their antivenoms to the WHO, and the WHO will then perform a really, really rigorous assessment of the antivenom. And they will independently test and verify antivenom manufacturers' claims of efficacy using the existing model. What they will also do is they will visit manufacturing facilities on site and make sure that the antivenom manufacturers are abiding to good manufacturing principles, so basically international standards of manufacturing. And the idea is, if they tick all these boxes, the WHO will give a stamp of approval, say this antivenom is effective, okay? And this will then hopefully give the assurance to ministries of health and procurement teams that the antivenom they're going to buy is actually effective for use in their country. So it gives you that bit, and hopefully will stop this use of ineffective antivenoms. Unfortunately, there have been a few unintended consequences, and I think this has been felt no more acutely than Kenya. One antivenom that's licensed for use, two antivenoms that are licensed for use in Kenya, Ainsworth and Vins, they failed pre-qualification from what I've been told is an administrative problem and problem in their manufacturing that they can address and then reapply to. But because they failed pre-qualification in this round, this led to the recall of these antivenoms across Kenya. They were removed from pharmacies across the country. And this left the situation where, for a very long time, there was just simply no antivenom because there was nothing to take its place. So that was a massive unintended consequence. I don't think that was ever intended by the WHO, but it caused serious problems, and it more than likely caused serious harm. But it needs to be, these things need to be thought through very carefully. So what are the alternatives? So, as I mentioned, this has been falling on academics a lot. Academics are seen as independent. They publish their results publicly so they can be independently scrutinized. And often they're the only source, other than the manufacturer's claims, on a therapy's likely effectiveness. This is showing that, this is assuming this, though, that we know what we're doing and we can be trusted, which is not the case a lot of the time, unfortunately. So what we did a few years ago is we reviewed all the preclinical efficacy data on antivenoms available in Africa since 1999, and this was up to 2018, so about 20 years worth. And we looked for papers that used a standard neutralization of efficacy assay, and we rejected anything that was an experimental antivenom. So these must be antivenoms that are available for treating people in sub-Saharan Africa. 18 publications met the criteria, which is not a lot. And what we found was we out of all the antivenoms available in sub-Saharan Africa, we found preclinical efficacy data, so mouse data on basically most of them. There was a few missing. Vinzekis is quite a new product. We couldn't find it at the time. We have done some preclinical data testing on this more recently, and it's looking pretty effective. Premium serum Central Africa, I think, has been discontinued. Same with Boomslang. There was actually no preclinical data for it in the normal ED50 model, but it has been tested in sort of variations of the model. And then we had this Barrett serum, ANZA antivenom D, which I hope just does not exist anymore. And Favafrique, which was discontinued. But even though we found preclinical studies on all of these, the amount of preclinical data for each of these individual antivenoms was found to be hugely variable. You can see here Echis ocellatus. So this is a heat map. The darker the blue, the more times it has been studied in a preclinical model. Echis ocellatus has been studied 8 times against against Equitab Plus. You can see Echis against across the board Echis ocellatus, which has now been renamed Echis romani, actually, has been tested quite a lot. And that's because it is seen to be the most medically important species on the continent. And then you get like Pulfider up here has been tested a lot against Equitab Plus. But basically, what you can see here is that these are medically important snakes, as noted by the WHO. there are a lot of snakes that have had hardly any preclinical testing done and made available publicly. And what you can see, so this is a graph that just shows some ED50s of the different antivenoms against different species. So we've got puff adders here, a few more of the Vitis genus, and then we're getting to Echis. Echis ocellatus, like I said, massive differences in tested lots. And there's huge differences in efficacy of some of the products, as you can see. So this is ED50 in mg per mil. These down here, the ED50 is about 1 mg of venom neutralized per mil, whereas up here, this tends to be more Equitab plus. These are, 4 times more potent. You're getting 4 mg venom neutralized per mil. Okay. For the vipers in the preclinical study, antivenoms tend to be much, much better at neutralizing lethality than neutralizing the lethality of the laphids. So what we also looked at is, because we know venom variation is hugely important, we also looked at where these preclinical studies were using their venom from. Were they specifying where did they get their venoms? And you can see anywhere on this map of Africa, anywhere with blue is where there's a preclinical study done from and reported. So Kenya's kind of well represented down here, Nigeria as well. But then there are huge swathes of sub-Saharan Africa with no data whatsoever. No data to demonstrate that the antivenoms that can be purchased, say, in Rwanda or in the DRC or in Ghana actually work against the venoms that are present in those countries. South Africa, I think, is a very, very odd one. No data for South African venoms at the time, considering Sama polyvalent is one of the best antivenoms you can use, and that's manufactured in Africa. And this is, like I mentioned, really, really important because of venom composition. So this is puff adder venom composition. This is a puff adder from Nigeria. This is a puff adder from Tanzania. The venom composition could not be more different. So there are substantial gaps in the data, but how good is the data that we have? Is it reliable? Is it reproducible? Can it be rigorously tested? And it's imperative considering preclinical murine models directly influence human treatment. So these have been published. So these are the ARRIVE guidelines. You can find them on the NC3R's website, and you can also just search for ARRIVE guidelines. These are it stands for the Animal Research Reporting of In Vivo and Experiments, and it's basically a reporting checklist. It's developed to improve reporting of animal experiments, maximize output, reduce numbers overall, increase reproducibility, which is really, really important, and prevent costly failures of drugs going forward. So we developed a sort of venom-specific ARRIVE checklist, looking at experimental setup, the animals, even what was the strain used, what was the weight, what was the procedure, and how well the procedures were reported. And it was very, very different. So, for example, only sort of 50% of the studies actually said what was concentration of the antivenom of the drug they were using. You cannot repeat that experiment if you don't know the concentration of the antivenom. Some of them didn't describe the antivenom batch, 30% didn't describe the antivenom batch, and obviously antivenom is a natural product. It varies massively from batch to batch. Only 60% of the studies actually said where their venoms came from. A good few of them had ethics statements, which is very important. And conflict of interest statements was only present in 60s. And the conflict of interest statement is really, really quite important because you've got to be very, very careful with academic publications. This is one of the publications in the study testing this product, antivitamin. The conflict of interest statement saying the authors declare there are no conflicts of interest, but 2 of the authors on the list are actually very, very high senior members of the manufacturing company of the antivenom. So I would say that is a direct conflict of interest. So you cannot trust that data very well, I don't think. Animals, very, very few reported on husbandry, very, very few reported on source, but the weight and the strain was much more better. Procedure. Some of the studies didn't even tell you what the injection route was, and the injection route pathology between an intraperitoneal and an IV route is massively different. It needs to be reported. But for most of them, they were well reported. And the procedural bits are really important, because if you get an odd bit of data in the data overall, you need to be able to report it. So this is all ED50s for neutralizing puff adders. And they're all fairly similar. But then you get this stonking big one out there that's saying it neutralizes nearly 40 mgs of venom per mil. Like, that to me looks wrong and not reproducible. And you cannot determine why from the data that was in the paper. Then we look at the doses effective, the units reported for the effective dose. There was 8 different units to describe the same thing. It was absolute. If you're a clinician trying to make heads or tails of this, or somebody in procurement, you 8 different units that are completely different. You're not going to be able to reliably dissect the data. So the conclusion from this is that preclinical efficacy reporting is a mess. It's currently impossible to perform any effective meta-analyses. It's impossible for clinicians to extract meaningful data quickly. And what's really important is it's really difficult for regulators to cross-check manufacturers' claims. So what we did was we've developed this sort of in vivo experimental reporting checklist, and basically putting out the very, very, very basics of things that every study must report in order to build this database of, this source of data, so that we can perform these meta studies in the future, which have been shown to be very, very effective in other fields. This is freely available. If you want it, you can Google it or just email me. I'll send you on one. So a sustainable solution, and this is what I'm really, really excited about K-Strek is doing, and the setup here is the sustainable solution. The only way forward is the establishment of expert antivenom quality control testing laboratories in sub-Saharan Africa. You need regional hubs to continuously test antivenoms that are coming into the country to assess their efficacy and their quality. The problem with the WHO preclinical guideline, preclinical evaluation is it's a one-stop shop. They're going to do it the once. It's not something that's going to be renewed going forward. So if an antivenom passes preclinical qualification now, it doesn't mean that same product will be preclinically effective 5 years down the line. It needs to be a continuous setup. So this is why K-Strek's setup here, I think, is really, really exciting, and is one of the most important sort of developments in snakebite research in sub-Saharan Africa in a very, very long time. So that's it. Thank you very much. I'm very pleased you've managed to stay with us for 1 hour and 45 minutes. It's been a slog. I'm hoping most of you managed to stay awake. Yeah, if you have any questions, please go ahead.
For a wonderful presentation, very informative, especially for those of us who are working with the Murine model. I felt like that was a crash course. You probably used the Murine model in our research, from the basics of animal care and handling, all the way to the interfaces of using them for drug testing and antivenom development. So thank you very much. So we'll take some questions. In the interest of time, we'll take the burning questions first, and then you can always approach Dr. Amy and Dr. Stuart afterwards. So any burning questions?
Thank you, Dr. Amy and Dr. Stuart. So I have a few questions. The first one, what's the proper unit of reporting an antivenom ED15? Because we find some papers reporting microliter per challenge dose, others the neutralization potency, which is milligram per ml. Yeah. And then we find others reporting actually the ED50 and ED100, just the ones that were found in the Murine models. So what would be the proper?
So this is an active and ongoing, I want to say discussion, but I think more of an argument in the snakebite community. There are proponents of each of the units, essentially. Some of the units are clearly not appropriate at all, because they don't really, they're not good in standardization across different antivenoms. In our preclinical review paper, we suggested milliliters of antivenom as the dose, because if you're using the milliliters of antivenom that you use to neutralize reflects what is being seen in the clinic. So if you're having to use a large volume of antivenom presumably in a human, you're going to have to use multiple doses, large volumes. The smaller the volume of antivenom you're going to use, the better. And we were thinking along the lines of trying to make it as clear and transparent to clinicians treating, this one is more potent than this one because this one needs 1 mil, this one needs 10 mil. Okay. The WHO guidelines, as they stand at the moment, they reckon there's 3 recommended units for publishing, which I don't think helps. So personally, what you mentioned, a lot of publications now have resorted to reporting their unit, multiple different units alongside each other, which I'm not sure is helpful. So it's something I think needs agreement at a global level. And I look forward to having that discussion with people. So personally, I use milliliters of antivenom just because I'm not the brightest and it's easy for me to decipher. But I think the mgs of venom per mil of antivenom is the one that's most commonly used, and we found that in our study.
And what would you have to say about those that report intraperitoneal LD50s and the intravenous LD50s?
So that is a problem, and because the way death is induced in those models are completely different. And with an intraperitoneal model, you need a lot more venom. And some centers, so ICR in Costa Rica, ICP in Costa Rica, sorry, they use an intraperitoneal model mostly, and they've done that for decades. We at LSTM, we use an intravenous model mostly. So it's really, really important to demonstrate, say, what the route of administration is, because the two of them are not comparable. And I think there have been cases where people have used an IPLD50 for an IV, and the mice, because it's such a large dose of venom, the mice have died immediately. So I don't think it particularly matters which route you use, as long as you are very, very consistent and very explicit in the route you're using.
And the last question is on the mice observation. Some, especially with the neurotoxic snakes, when you inject a mouse with a neurotoxic venom, you'd see lethal signs in the mice. So what's the appropriate time of maybe working on humane endpoints? And the times when you consider the humane endpoints, you'd find the mice struggling the first hours and then surviving. So you might induce a humane endpoint and end up reporting this as mortality, but that would have been survival.
Yeah, and this is a big problem. It's a huge problem. So any humane endpoints you implement, you really have to be sure that they are they can stand up to scrutiny, essentially. So the humane endpoints that we've developed, so we shortened the time to 6 hours so the normal model is 24 hours. We shortened it. So the first thing to do was shorten it to 6 hours, because we demonstrated that if a mouse was going to die, it was probably going to die within the first 6 hours anyway. Very few mice died after that. The humane endpoints, you need to do those pilot studies, and we have to explicitly say in our license, if we're putting in a humane endpoint, especially in terms of envenoming, where the mouse model is all that's used before it goes into a human, we have to be really, really sure of those humane endpoints. So in the model as it is, I would say the humane endpoints that we use, they're really reliable, depending on the species that you're using. But you have to be confident and have to show that they're reliable as well. You can't just basically say, oh, this mouse probably won't survive the 24 hours. You've got to be sure that if I left this mouse, it would die. I'm hoping that with the newer models we're developing, that kind of haziness, uncertainty will be much more clear, and we will be able to, because we're using biological markers, not just life death, which is a variable thing anyway. There's so many venoms are so variable, especially in viper venom is really, really difficult to work with. Because we're having a very clear readout of the biomarker, we're hoping that we can demonstrate very, very easily that this is a humane endpoint. So what we're doing at the moment for neurotoxicity, we've not started yet, but we'll be starting soon, is we're looking at sort of vital signs. So we're going to be measuring heart rate, O2, O2 saturation. We're going to start doing non-recovery experiments, so we can do ECGs on the mice while they're being envenomed as well. So we're hoping to build up a really comprehensive picture of what happens to the animal during envenoming, and not just this animal looks sick, it's going to die. Okay.
Any other burning questions? Comments?
Thank you for that great presentation. In your presentation, you mentioned that there are few clinical trials that have been carried out. And again, you say that in a study like this, you cannot have a placebo because you cannot give somebody the venom and then you fail to treat the venom. So I just wanted to know, were these studies randomized? Because if you look at if you're looking at the evidence of effectiveness of an intervention, randomized controlled trials are actually at the top. On the same line, again, you mentioned that there's the possibility of doing clinical trials in future. And I don't know, snakes are really feared. I don't know how you're going to convince the participants of the studies that you're going to include in the studies that I'm going to give you a snake venom and then try to give you a treatment.
So it's a really, really good question. So the clinical trials that were performed, they were all randomized, double-blinded control trials. So people were offered a treatment regardless. And then, yeah, I think it was a reduced dose on top of the antivenom that was given. What we're doing is we're looking for sites to perform clinical trials where there's a really high incidence rate of bites by particular species. So northern Nigeria, where the previous clinical trials were done, would be perfect for, Echis bites, because you get huge numbers of bites. And because the snakes are really feared in those areas, and there are already existing specialist snakebite hospitals, the local communities are actually quite well educated on what's doing in terms of snakebite, and they will go to hospital themselves. So recruitment, yeah, it probably is going to be challenging, because you've got to take everything into account. The patient's panicked. They might just say yes because they want to get the treatment very, very quickly. So you need to put in safeguards, like they can withdraw consent afterwards if they've managed to calm down a bit. So yeah, it's not my area of expertise, but there are a lot of safeguards put in. We're also looking in sort of areas of northern Brazil as well, where you get a lot of Bothrops bites. We've got hospitals there that we're identifying, East Watini as well. So the clinical trials in the future, from what I understand, they're going to be fairly similar. Dr. George can probably tell you much more about these because he's helping design them. But what I would really like to do in the future is human challenge models with venom. And that's persuading people to voluntarily have venom injections. This is the reaction I get everywhere. But I've been talking about this for about 5 years now, and people are starting to come around to the idea. So I believe in, say, 20 years' time, human challenge models of envenoming may be more commonplace than. And this is on the basis that at LSTM now, we've just got a new facility. We've got a human trials center basically, and a human challenge center. We've got the biggest inpatient research facility in the UK, and we're going to be doing human challenge models. We're already doing them with things like tuberculosis, malaria. So people are offering to be deliberately infected with TB and malaria, COVID, those kind of things. So somebody put it to me the other day, I'd rather have an injection of snake venom than get deliberately exposed to TB. So I think you could probably persuade people to do it. Yeah. I'll send an invite out looking for volunteers if it ever happens. I think we've got one more question.
Yes, one more. And we also have to acknowledge our online audience as well. There's one question on YouTube, but we'll start with Dr. Atunga first.
Thank you very much for the nice presentation. I think it was really interesting. Thank you. I just have 2 questions. One is on the 3R. I see a lot of people are adding more Rs I
Really?
think they are adding more R's,
Mhm.
like responsibility or rehabilitation. You know, they're
Yeah.
adding more R's. Eh?
Yeah.
So maybe you can comment about the additional R's that are people continually adding. I know the 3 R's are the original, are the foundation of everything that we do as far as animal
Mhm.
work is concerned. So maybe you can comment about the other R's. My second question is about the snake antivenom. I noted that most of the studies, preclinical studies, are using rodents like mouse models. Maybe you can comment about the other models like rabbits or other models. I don't know whether some people are using these other models to test the efficacy of of new antivenom. Maybe you can also comment on that.
Sure.
Thank you.
So the first one with the extra R's. So the additional R's like rehoming, rehabilitation, they are, I think, and maybe Amy would be better placed to answer this. I think they're being driven by needing to reflect the public's attitude on animal research in different places. So rehoming of animals, especially protected animals. So in the UK, a protected animal, especially protected animal, is usually companion animals. So like horses, dogs, cats. Those kinds of research are really the public's opinion on that kind of research is very, very strained because they see them as companion animals, which they are. So rehoming in those kind of senses is really sort of encouraged where possible. I think the original 3 R's, I think, are the most important. Refinement, I think, is definitely the most important because I think at the moment in time, technologically, complete replacement of mouse models or animal models, I just simply don't think it's possible. I'd like to say it is possible in the near future. I just don't think it is. Being a pragmatist, I don't think it is. All the extra R's at the moment, they can be suggested, but they are not in the UK anyway. They're not enshrined in law as it is. So theoretically, you can implement them if you want to, but you're not under any requirement to do so. So that's that. Other mouse models. So rabbits, the main reason I don't think rabbits are used often is their expense, especially in the UK. We used to do a lot of rabbit testing at LSTM in the 80s, but now rabbits, how much does a rabbit cost now? I think a lot more than a mouse. I think it's about maybe £60 a rabbit, and then you've got to pay for its maintenance as well. So it's incredibly expensive. Also, they take up a lot more space within the animal units because they've got to have pens on the floor. So I think it's just cost and what's the word I'm looking for? Practicality is why a lot of models aren't used. Also, the other reason is in UK law is, and in European Union law is you have to use the least advanced species, basically. And a mouse is considered less advanced than a rabbit. So if there is a less advanced species that can be used, you have to go down to the rabbit model. Mouse model, sorry. Okay. Thank you very much.
We have one from the YouTube, and he says, that is John Kagira. Congratulations to Catherine. It will help a lot in quality control of antivenoms. And then he says also, good work. Can the SCID mice be used for all valvulas? The SCID are quite expensive to maintain, so we might still need other types of mice.
This one's for Amy.
Yeah, that is the problem with the immunodeficient mice. They are very, very expensive. So there are different strains, which is where you sort of weigh up, that sort of thing at the same time. So your experimental model and the cost as well. But yeah, they are used for ovolvulus as well. But yeah, that is one issue is the expense there, which is sort of the cost benefit of, the mice are getting parasites. you know, from those models compared to a wild type where you don't get as many, where you might have to buy more mice. So it's sort of weighing all that up, really. I hope that answered some of it.
Then last but not least, I just want to know your future plans for the antivenoms in sub-Saharan Africa. Thank you.
So in terms of the research we're doing, myself and Amy doing at the moment, we're really focusing heavily on the development of new models so we can enable that translation better and support clinical trials as well when they eventually happen. Antivenom development. So another project I'm working on is there are lots of shortcomings with antivenoms. The way they're made is very, very crude in that you take crude venom, which is a complex mixture of up to 100 different proteins, all with different pathologies, all with different toxicities, all with different immunogenicities, and you inject an animal with it. And you hope that the animal generates antibodies potently to the medically relevant toxins. What actually happens is some of the medically unrelevant toxins can dominate the immune response, and you generate a lot of antibodies that are clinically redundant, okay? And this is one of the reasons why you need to give really large volumes of antivenom to people when they're bit. What we're looking at the moment, so at LSD at CSRI, we do research really across the translational spectrum. So myself, I'm working more on conventional antivenom development. We're working on next generation therapies, so monoclonal antibody therapies, recombinant antivenoms. We're also working on antivenom alternatives. So we're doing a lot on small molecule drug therapies. We've actually got a clinical trial down at Kemry in Kalifi, I think it is, on a drug called unithiol, which is a heavy metal chelator that looks really good for hemotoxic envenoming. And we're also trying to, you know, push forward clinical trials and better regulation of antivenoms. So we're working across that sort of spectrum. What I'm doing is I'm trying to replace, I don't think Joffrey would enjoy hearing this, I'm trying to replace the use of crude venom in antivenom manufacture by rationally designing synthetic molecules that look like venom. So you can choose what you're immunizing the animals to. So you can focus it on the clinically relevant toxins. And you can do it in a way on a carrier molecule, such as a virus-like particle, that really stimulates the immune system to produce very, very potent antibodies. So all we're hoping to do is just simply increase the efficacy of conventional antivenoms. My focus on that is very much on sub-Saharan African neurotoxic species. So your mambas and your neurotoxic cobras. So, yeah.
Okay, thank you so much, Dr. Stuart and Dr. Amy, for addressing everyone's questions. We've run out of time. Is it a brief question, or are you able to ask it after?
I have a question for Dr. George and the Kenyan team. Are you guys working on the antivenoms that work on the snakes in the country? Are you at KISRIC working on antivenoms for the Kenyan snakes?
I'm not really getting your question, but so yeah, we are focusing on Kenyan snakes. So we extract venom from Kenyan snakes, but the plan has always been to expand to East and Central Africa. So we have some actually snakes from Tanzania and likely very soon from Uganda as well. So the antivenoms we are likely to generate from the work we're doing, of course, will capture the range of that. But just like Dr. Stuart has said, we are working on new generation therapies for treating snakebite or antivenoms, the monoclonal antibodies, the synthetic ones, etc. And this likely, because you just don't want to capture Kenya, the numbers are not so many. So the idea has always been to capture sub-Saharan Africa, and we have India on board as well.
Thank you. Thanks, everyone. Feel free to approach Dr. Stuart and Dr. Amy after the presentation. So another round of applause for Dr. Stuart and Dr. Amy, please. We'll now get a vote of thanks from our Chief of Research, Dr. Atunga, followed by some closing remarks from Dr. George.
Good afternoon, everyone. Thank you for your patience. I note that I think today we have sat a bit longer, and probably because the presentations were more interesting. So we normally spend 1 hour, but for people to stay more than 1 hour, I think you must have interested them. So probably they want to follow what you have presented, and maybe some of them are aspiring to do studies. Some of them are students, and some of them are staff who want to do further studies. Probably they want to do snake, or what was the other disease she presented? Was which was Vilaria, something Vilaria. Yeah, and what else? I think I saw snakes, Filaria, isn't it? Heartworm. It's called heartworm. Oh, okay. And then, of course, animal welfare. I think people study animal welfare, isn't it? They do issues, masters on or ethics and so forth. So those are areas that you can explore. Mine, the director is not here today, and you know he already went because of the staff who had a challenge. And I know some of you are leaving, also going there this afternoon. But on behalf of the director, I want to thank the presenters. Thank you very much for coming. And thank you for, again, agreeing to give a presentation. You know, sometimes you can come and decide not to present. So we are really grateful that you decided to come and spend your time to make presentation to the audience at IPR. And George is the one who persuaded you. I think that's very good. And I know we have a very good relations with the UK. I know LSTM has been very supportive on issues of snake, but we also have other collaborators. For example, University of Leeds, we collaborate on issues of animal ethics. So that's why we are also very passionate, because also this is an animal facility. So we ensure that actually we are there to the 3R plus many Rs that are coming on board. But thank you very much. And we wish you a good stay in Kenya. I don't know whether you are going very soon or you are staying here longer. That's the you leave. So, okay, within. Oh, that's good. And I hope some people will have more time to interact with our visitors. They will be around until Thursday. So meet them, talk to them, share your interests, and make friendship. I think that is very, very important. And what I can say is that when you go back to the UK, pass our regards to everyone there, and also come again. So thank you very much, and I wish you well. And everybody else, I wish you a nice afternoon and enjoy your stay. Thank you.
Okay.
Normally, you don't talk after the pause. So.
Just a bit of housekeeping. Thank you, Dr. Atunga, for the vote of thanks. And thank you, everyone, again, for your patience, for your attendance, and for your level of engagement. Tomorrow's seminar has been pushed to Thursday at 11:30. So we have